So many thanks to Sarah for the kind invitation to chair the session. It was on the condition that I'm relatively polite, I think. So I will do my best. It's a pleasure and of course an honor to introduce Professor Syma Khalid for her research seminar today. So just a quick bit of background. So after obtaining her PhD with Mark Roger, I first met Syma and got to know her in Mark Sansom's lab when she did her first postdoc. I think 2003-ish, 2004, something like that. Yeah. And I think it's fair to say we've been great friends and collaborators ever since, through our mutual interest in bacterial membranes and also our fellow suffering as Liverpool supporters. Yeah, after her postdoc, Syma's career progressed really rapidly. She quickly established a hugely successful research group at the University of Southampton. And I think it's fair to say that she is the world leader in multi-scale simulations of bacterial envelopes, membranes, and cell wall components. I'm also very fortunate to be a long-term collaborator of Syma's, and when she's not watering her pumpkin patches in Animal Crossing, we are co-supervising several PhD students together, and that's, I think, resulted in lots of exciting discoveries as well. So I'm very grateful to have been involved in all of that. So without further ado, I will hand over to you, Syma. And I think your talk today is entitled "Towards a Virtual Bacterial Cell Envelope, Adding the Biological Complexity to Biomolecular MD Simulations." So over to you.
Cool. Great. Well, thank you very much. Thank you again to both Sarahs and also to Pete for the very generous, largely generous introduction. As he says, we've known each other for a long time. Okay, so I'm just going to share my screen. Right, can everyone see that? No?
I can't.
Okay.
Yes.
Okay.
Yeah.
Okay, cool. So let me just start with that. Okay, so I'm going to start off with just 3 slides. Firstly, discussing with you and describing to you HEC BioSim, just because it's a sister organization of CCPBioSim. So I'm the chair of HEC BioSim, which is a UK-wide consortium that promotes the use of sort of very high-end computing resources by the biomolecular simulation community. We're an inclusive open group, and we started off really by providing time on the national supercomputer called Archer, which I've no doubt many of you will have heard of. But in recent years, I guess really over the last 18 months or so, that's also begun now to incorporate the so-called tier 2 machines. And I'll describe on the next slide what I mean by tier 2. So basically, there's a bunch of resource that we have that we can provide for biomolecular simulation research. So for example, if your local university cannot provide you with the resource you need, you don't despair. Likely we can do something to help. But not only do we provide hard sort of time on these supercomputing facilities, we also provide support, help you with best practice, and also expertise with if you've got a favorite code that you want to use, how you might want to port that over for high performance computing resources. So we've got lots of technical support we can provide too. I've given at the bottom there the URL for our website, which does contain a whole bunch of information. Just give you a snapshot from that website here, where we have online forms where you can apply for time on Archer. You can apply for time on Jade at the moment. Jade is a GPU cluster, which is largely used for machine learning, but there's an element of it which is reserved for biomolecular simulation. So if you apply, if you have a code that's optimized for GPUs, then actually working on Archer, which is CPU based, may not be the best thing. You may prefer to go for a GPU cluster. So it may be worth you talking to us about applying for time on Jade. I've given you there a little snapshot of the management committee group. the management group is actually built up of established biomolecular simulation groups from across the UK. And the way we've put together the group is to try and represent the users of the popular molecular dynamics codes, people who apply those codes in a range of different ways. So for example, there'll be applications that are very capability based, and there's others that are very capacity based. So for example, there's those of us within the consortium who like to simulate very large systems for long simulations. But then there's others who do lots of simulations of smaller systems, but they also need loads of resource, but just because of the sheer number of simulations they run. And it turns out that different architectures are suited for different types of calculations. And so the management group represents as much of the breadth of the field as we can possibly get. I've highlighted in particular Dr. James Gebbie-Reyett, who's our COSEC support. So he's based over at STFC. So James is really, I guess, the heart of HEC BioSim. He's able to do benchmarking calculations. He provides support with porting code over, helps. He's developed, I've just given a little snapshot. It's a little bit small to see, which I apologize for, this thing called the HEC time calculator. So this is an applet that James has developed, whereby the user can input the size of the calculation, the type of calculation they'll run, the code they would like to run that on, and it provides a good estimate of the type of resource that will be needed, which is then needed to input into the application form. So James, in a way, I mean, I guess sort of my name's on the grant, but really James really runs HEC BioSim. And so he's usually he's certainly the first port of call for me when we get queries. And finally, the last slide is I just wanted to provide a brief overview of what's coming ahead within the UK landscape for high performance computing. And I've called it exciting times ahead because it really is, and things are changing. So for example, the national facility, Archer, is right now actually in the process of being upgraded to Archer 2, which will give us a huge boost in terms of capabilities. But also alongside that, so that's the national facility is tier 1. Tier 2 is the level that sits beneath that. And these are smaller machines, and there's a whole range of them around the UK. They're smaller than the national facility, but they will be larger than your local university machine. And the second round of these machines are now beginning to come online. There are a whole range of architecture. So there's some based on CPUs, there's GPUs, there's ARM architecture, there's some that are hybrids of the different, including different components. And the way things are moving is that in future, it will simply be a case of matching up the type of calculation you want to do with the architecture that's best designed for that. And so how that moves forward in terms of what do you, the user, have to do to apply for these resources is still being worked through. And I urge you to keep an eye on the UK RI website and also the HEC BioSim websites for updates. We'll be providing, I'm on various EPSRC panels alongside being on HEC BioSim. So we are in a good position to keep our community updated. So just for completeness, and context, so tier 1 is the national facility. Tier 2 are these additional machines that sit somewhere above your local university level, where tier 3 will be your local university machine. And of course, not all universities have access to the same facility, and that's why it's really important we provide access to tier 1 and tier 2. But above tier 1, there's also tier 0, which is sort of at the international level. For example, you can apply for time in PRACE in Europe, or Insight in the USA, where they use various Department of Energy and Defense machines. So I guess the bottom line is that as we move forward, there's a whole bunch of resources available to you. If your university machine is not providing you with the resource you need to run the calculations you want, please don't despair. Get in touch with us, keep an eye on our website, and we'll be happy to answer any questions and to help you sort of find the right machines, the right architectures for your applications. Okay, I think I'm going to stop talking about HEC BioSim there, but I guess could we take questions on this part now, maybe? Yes, no?
Yes. People want to ask questions? You
can... Yeah. There are no questions as yet, but
okay. Well, maybe that gives people a chance to think about it, and they can ask them all at the end then. If
yeah.
Shall I just move on to the science part? Okay.
Yeah, I think it makes sense.
Okay, cool. So, folks, if you have questions...
Sorry. There is one question now if you want to take it.
Yeah, sure.
Is there any European-wide project?
So there's PRACE, right? So PRACE provides access to resources across a bunch of machines within Europe. So to be honest, I personally have never applied for PRACE time, but I know many people who have, and many of those machines give bigger chunks of resource than Archer will. So there's Mare Nostrum in Spain, for example. I think they have a more formal application procedure than we have for Archer, but there's a lot of help available. And my understanding is that so the UK involvement with PRACE will not end as we Brexit. Okay, so take a look at their website. That project is there within Europe. And of course, there's other things like, just trying to remember the name of, there's a scheme whereby you get access to a European machine, and UKRI also provide resource for you to go. That's not you, it's combination of UKRI and EU, where you can then go and actually spend time in the country where that machine is based, and they'll pay your living costs as well. I'm just trying to think of the name of that scheme. Sorry, it's just completely gone out of my head. HPC Europa, that's right. So there are projects like that available.
Yeah, and I think it's definitely worth contacting the people. Go to the website and contact them, because even Singapore, there are various schemes, and even Singapore has been able to access computers via PRACE in some situations. So it's certainly worth talking to them, I think.
Yeah, absolutely. I mean, these machines are there, and they're there to facilitate good science. So if you have an excellent proposal and you feel you don't have the resource available locally, then please do contact people.
Yeah, that's right.
This day and age, that shouldn't be something that limits research.
Yeah. We do have one other question,
Sure.
which is HEC BioSim open to all universities or just universities within the UK?
I'm afraid it's open to just universities in the UK.
Okay.
But I think, for example, Insight in the US is open to any country, as far as I know. So I used to be on the panel for that actually a few years ago now. But certainly that was not just for, I mean, the machines are provided by UK Department of Energy and Defence, but nevertheless, the resource is available for non-US people.
Okay. Yep, I think that's all for the questions for the moment.
Okay, great. Right, let's move on to the science. Okay, so in the next, I guess, 35 minutes, 40 minutes or so, I'll be discussing some work we've been doing in my group for a number of years now. I guess pretty much from when I really set the group up in about, I'm trying to think when that was, about 2007. So it's sort of projects that have developed over time. And what I'd like to discuss with you in particular is, going through this all the time, is when we do biomolecular simulations, sort of asking ourselves why we're doing the simulations, what we hope to get out of them, and how do we interpret the results. Well, I certainly believe we can over-interpret results, especially when the systems we're looking at are biological, because there's so many variables and there's so many things we don't yet understand. So some of this is inevitably there'll be a few sort of success stories, but littered in within that will be sort of projects where we went off the wrong track, and trying to sort of give lessons where I hope others will learn from and not make the same mistakes we made. A quick biology lesson. I hope Peter's listening in particular, because he's a bit hazy on this stuff. So the E. coli cell envelope, it turns out certainly for so because E. coli is gram-negative bacterium, the cell envelope is quite complex. And for me, this is of endless fascination, given bacteria are single-celled organisms, and we, of course, have millions of cells. These bacteria still manage to evade all of our pathogenic bacteria, evade all our attempts to try and combat them with antibiotics, for example. Right? They're still managing to somehow come up with resistance mechanisms. And much of this comes from the complexity of the cell envelope. So you can think of it as a sort of sandwich where you have two membranes. So there's one that's on the outside, that's the sort of the first physical barrier that's encountered by anything that tries to enter the cell. And then you've got the inner membrane, which is on the side of the one that's just that's sort of connected to the cell interior. And in between, and if you think of those membranes as the bread of the sandwich, in between the filling is an aqueous region, which is called the periplasm. Now, the outer membrane is enormously complex in that it's asymmetric in terms of its lipid composition. So we all know that biological membranes have two layers or two leaflets with lipids. Well, in the outer membrane of gram-negative bacteria, we have a very complex molecule called lipopolysaccharide. It can have between 6 and 4 hydrocarbon tails. It's connected by head groups which can have varying amounts of phosphorylation, and then above that has levels and lots of levels of sugars. And so in a way this molecule encompasses pretty much all of chemistry. It has hydrocarbon regions, it has charge groups in the phosphates, it has chirality in the sugars. Of course, also in the sugars, it has the capability of hydrogen bonding. And so there's a lot of chemistry going on, and ignoring that means that all this important chemistry is ignored. The inner membrane is, by comparison, more simple. It has these phospholipids on both sides. And of course, there's proteins jam-packed in both membranes. The region in the middle, the periplasm, as I said, this is aqueous, but it contains what's called the peptidoglycan layer. This is also called the cell wall, and this provides a sort of skeleton, if you like. It gives rigidity to the cell. So this arrangement of the two membranes and the peptidoglycan, it surrounds the entire cell. Okay, so you have to breach this defense if you want to gain access to the inside of the cell, which many antibiotics do. Antibiotics work by either breaking the cell envelope and destroying that, or by getting through it into the cell and interfering with some process inside the cell. And before we go on to talk about the simulations we've done, I just wanted to put this slide up here, given this is a training week, to ask, well, why do we want to do simulations? Clearly, one of the most obvious reasons is that we want to rationalize and understand at the molecular level some very specific experimental observations. Often experiments can't reach the molecular or the atomistic levels. They get some results and then call upon simulation experts to help understand and rationalize why they're seeing those results. It could be that we want to study some system under conditions that are difficult or expensive to achieve using experimental methods. So difficult to achieve pressures or temperatures, for example, and it's cheaper and easier to use computational methods. But I think increasingly important is that actually we want to develop and put forward new hypotheses. We want to do curiosity-driven science. Just to ask the question, let's see what we find. And I think that's perfectly okay. Coming from a very traditional chemistry background, we're always told that an experiment is done to test a hypothesis. But actually, I think experiments can be done to put forward new hypotheses. I mean, many of us are here doing science because we were the kind of kid who would pick up the pebble to see what was underneath, right? We didn't know what we would find. There wasn't a specific question that was being asked. And so there's this idea and to put forward new hypotheses where you don't actually have a clue what you might find, and you do the simulations to get to that point. And that leads us to another point, which is it's equally important for us as simulation people to drive projects, for us to come up with the hypotheses that we then give to our experimental colleagues and say, you do the experiment to prove me right or wrong, or to tell me why this is happening, rather than it always being this other way around where the experimental guys go and do the experiments and then come to us and ask us to rationalize. And I think as computing power improves, we're increasingly in a position where we can lead these projects. Okay, so back to the outer membrane. As I mentioned right at the start, this molecule LPS, which sits on the outside of the outer membrane, is horribly complex. It has these hydrocarbon tails, this sugar, you can see just one here in the gold and red, which has got a phosphate group, and then levels and levels of sugars. So when I started my group, there was only one model available for LPS, developed by Tjerk Straatsma and Teresa Suarez. This was using the amber force field. The sugars were very accurate, but I think it's fair to say there were some inaccuracies in the tail group. At that time, my group, and independently of us, Wampil Im and Jeffrey Clowder started developing models of LPS. Im and Clowder used the charm force field, whereas in my group, we use a Gromos force field. And what happened actually is, I think, very nice, is we're at a stage now where the amber guys have also fixed the issues with the lipid tails, such that there are now charm, amber, and Gromos force fields available for LPS, and they by and large agree very well. I'm just putting up this one image here of some work we did with Jeremy Lakey's group in Newcastle, and I hope this will convince you that our model is pretty accurate. So Jeremy did some experimental studies where they used chelating agents to remove divalent cations from the head group regions of these LPS molecules. So it turns out that LPS is held together by calciums and magnesiums in the head group region. Okay, so they ripped out those divalent cations and replaced them with sodiums, keeping the ionic strength the same. What happened was the membrane just lost its integrity, no longer formed a nice double layer. And indeed, the same thing happened with our simulations. You can see here that in just 200 nanoseconds, when we've removed the divalent cations and replaced them with sodiums, can you see here that the water shown in cyan is beginning to seep in, and we no longer have a nice double layer. And I think this is a fairly convincing pictorial argument for the model being quite accurate. I won't bore you with all the sort of more quantitative models we have. Okay, so that was all well and good. One of the first things we did, as I said, is we developed this model. But a model's no good unless you use it to learn some new biology. And so to do that, we turned to our favorite protein, OMP. This is a very well-studied outer membrane protein. I'd done simulations on this when I was a postdoc. Our chair, Pete Bond, did simulations on this for his PhD, so we knew this protein very well. It turns out that despite it being studied a lot, there is some controversy in the literature of what its oligomeric state is in vivo. Is it a monomer? Is it a dimer? And indeed, there are still arguments out there for higher oligomers. Our collaborator at Oxford, Carol Robinson's group, did some mass spec studies, and where they found evidence for dimers of the full-length protein. So here, I'm just showing you the bit that sits in the membrane. Okay, there's a transmembrane sort of barrel that sits in the membrane, but actually it turns out that the full-length protein also has a large linker region, and then a soluble domain that sits in the periplasm and crucially also non-covalently binds the cell wall. So we thought, well, this has been looked at by mass spec studies. it would be useful for us to have a look at this in context with the membrane present and the cell wall to get some insights into how it binds the cell wall. So luckily for us, there was a crystal structure of the C-terminal domain, albeit from a different organism, of this protein bound to a small portion of the cell wall. Should just say here that the cell wall is composed of, it's a polymer composed of repeating sugar and peptide units. So we built in the missing bits just to give us a complete monomer of cell wall. And we ran some simulations. In particular, the crystal structure showed 2 salt bridges, so 2 electrostatic interactions between the protein and the cell wall. And we found, as we ran our simulations, we picked those interactions were very, very stable, which gave us some confidence that, the simulations were accurate and we were binding the protein in the correct regions. Interestingly, we found that as we ran the simulations of the monomeric protein, after a while, the linker region of the protein contracted, and the C-terminal domain, which we know is bound to the cell wall, and remember the cell wall is a big sheet that covers all of the bacterium, this small portion of the cell wall that we had was being lifted up such that it was interacting with the lipid head groups of the membrane. That's just puzzling because we wouldn't expect this to happen. We know from tomography data what the separation is between the membrane and the cell wall. What we were seeing would lead to distortions of the cell wall. Interestingly, when we ran simulations of the protein in its dimeric form, we did not see the interaction of the cell wall with the membrane. And the reason for that is that the dimeric interface the dimer is holding these C-terminal regions together, so they can't lift up individually and interact with the membrane. So that led us to, well, hypothesize that, well, could it be that according to our results anyway, when you have the protein in its monomeric form, you see the cell wall being lifted up and interacting with the membrane. So if it was indeed a big sheet, you'd get something that looks like the schematic diagram on the left there, a huge distortion in the cell wall. Whereas when we have the dimer, we don't have the distortion in the same way. So what does this mean? Does it mean that binding of peptidoglycan by the monomer in real life causes severe distortions of the cell wall? Does it mean that OMPADE, when it's in its monomeric form, doesn't bind peptidoglycan at all? I mean, looking at tomography data, we hadn't ever sort of seen such big undulations or such big distortions of the cell wall. So it was difficult to rationalize or to really that the protein would be causing these severe distortions. But of course, in the back of our minds was always that, is what we're seeing an artifact of the fact that we've used really short strands rather than a large sheet of the cell wall? And I guess this takes me back to what I said right from the start, where it can be easy when doing simulations of biological systems to overinterpret what one sees. So we'd taken a tiny portion of a huge polymeric mesh and we'd simulated it, and we were seeing results that were unexpected. It's tempting to say that perhaps we're seeing distortions of the cell wall, or when it's a monomer, it doesn't bind the cell wall at all. But we have to bear in mind that what we simulated was not particularly realistic at this point. We then moved on to make a bigger model of the cell wall. So now we did indeed make a big mesh, and we bound it to itself across periodic boundaries. So it is actually a continuous two-dimensional structure, if that makes sense. So it's a flat structure, it's continuous in 2D. We set the system up where we take a monomer of the protein, and we started the simulations by placing it at about 90 angstroms from the cell wall. We took that distance from an estimation from cryotomography data. We ran the simulation, and frustratingly, nothing happened. Right? We didn't see any interaction of the protein with so we didn't see any interaction of this bit of the protein with the cell wall at all, even though now we've got a more complete model of the cell wall. But then when we started thinking about it, we realized that we had made a big omission. We'd missed out this protein here called bronze lipoprotein. Now, this is the most abundant protein in E. coli. So there's shed loads of this everywhere, and we hadn't put it into our simulations. So when we went back into the simulations again, now adding in bronze lipoprotein, what we saw was that this protein, sorry, I should say this protein is covalently bound. It's the only protein known that's covalently bound to the cell wall in E. coli. Okay, so it's linked to the cell wall covalently. Those bonds don't break. On the other side, it's anchored via lipidated regions into the membrane. As we ran our simulation, we found that bronze lipoprotein was able to tilt a little bit. It was able to kink a little bit. And in doing so, it led to a reduction in the gap between the membrane and the cell wall. So, between the protein OMP and the cell wall. And spontaneously, the protein sensed the cell wall via electrostatic interactions. The linker region expanded, and the C-terminal domain of the protein landed on top of the cell wall. And rather pleasingly, it picked out the same electrostatic interactions that we'd seen in the crystal structure being really important. Right, so that was a really, we felt a nice result. By putting in the bronze lipoprotein, this crucial ingredient, we'd shown that OMP as a monomer does but indeed bind to the cell wall, and we did not get that big distortion we were seeing when we only had tiny portions of the cell wall. Intriguingly, however, when we looked at the simulations of the dimer didn't seem to need bronze lipoprotein. It could by itself just, if you like, drop the anchor of the C-terminal domain onto the cell wall. And I can only rationalize that as maybe now you've got twice as many electrostatic interactions as you have in the monomer, and there's some kind of threshold level. And so that's enough for the protein to sense electrostatically the cell wall and change its conformation to interact. And, I guess one's tempted again to maybe make a hypothesis that could this be a in vivo role for the dimeric protein, that maybe when the absence of bronze lipoprotein, having these two monomers together provides a tight interaction with the cell wall. But it could be that, we're missing something else in our simulation. So again, I'm hesitant to say that with any kind of certainty. So at the moment, we've looked at half of the sandwich. We've looked at the outer membrane and the cell wall. But what's still missing is the inner membrane and any proteins in that membrane. So just to complete the picture, we took a protein called TOL-R, which sits in the inner membrane and has been shown to bind to the cell wall. The protein binds to the cell wall in what's called its open state, but the crystal structure shows a protein in the closed state. So but there was a model of the protein in its open state made by Erin Cutts in Oxford. So we asked if we could take a look at this, and they didn't run any simulations of the model. They just produced the models. So we asked if we could test that for them, which they were very happy to give us the model. So we ran some simulations, and these were the first ever reported simulations in which, if you like this, you've got a little portion or a chunk of a virtual cell envelope. We have the outer membrane, the inner membrane, the cell wall, and proteins in both sides. And the sort of actual more detailed image is shown here, where you can see the proteins in a bit more detail. All I've done here is plotted in the Z coordinate of the center of mass of the protein OMP in the outer membrane in blue, the cell wall in red, and this TOL-R protein in the inner membrane in green. And you can see there's three different simulations shown here. This is why there's three curves for each color. You can see that over these simulations, they're fairly stable. They don't move. We're not seeing any distortion of the system. They seem to be pretty happy in their orientations. And indeed, what was really nice was we were able to identify residues in this TOL-R protein that are key for binding the peptidoglycan. And these are the same residues that have been shown to be important by the experimental groups. So again, the simulations and the experiments are agreeing, which is nice. But I think perhaps what's nicer is we were able to show how the binding occurs. So when we ran the simulations, we placed the protein just beneath the cell wall. So it wasn't interacting. As the simulation progressed, we saw these terminal regions of the protein, sort of, if you like, almost reach up and grab hold of the cell wall and then pull the rest of the bulk of the protein up a little bit so they form their full complement of interactions. So this had never been seen before, right? The experimental guys knew which amino acids in the protein were important for binding to the cell wall, but they'd never seen the process of binding, which these simulations showed. So they sort of agreed with the experiments, but added on something extra. And interestingly, we found that if we look at the morphology of the cell wall itself, when we only have the protein on paybound from the outer membrane side, you can see you've got a slight distortion in the cell wall. I mean, nothing like we were seeing when we only had these short regions, but nevertheless, it's not flat. And equally, if only TOL-R, so the protein from the inner membrane side, is bound, again, we see a slight undulation in the morphology. When both proteins are present, we get a really nice flat cell wall. And so again, we're tempted to say, but we do so hesitantly, that it could be that at any given point, we know these proteins are diffusing around laterally in their respective membranes. When you get a point at which proteins from both sides are evenly distributed, you get a nice flat cell wall. But if there are regions where they're not distributed evenly, and that can happen transiently, it would lead to some slight distortions, which over the fullness of time would likely even out. And so I think now we have both membranes, we have the cell wall. So we were pretty pleased that we've got this portion of a little portion of the virtual cell envelope. But I guess one could ask, well, is anything still missing? And it turns out there is. It turns out that the crowding is missing. Right. So far, we've only got one protein in the outer membrane. We've got one in the inner membrane. The periplasm, which contains the cell wall, that aqueous region, it too is jam-packed full of proteins. Indeed, in the past, it's often been thought of as being in a gel-like state. And we haven't included those. All we've included is sort of the big structures. So now this is unpublished work. It's under review at Structure at the moment. We've built a system where we've put, again, OMPE in the outer membrane, and Blonde's lipoprotein is in, of course, but now we've put some additional proteins into the periplasm. A couple of them, so PAL and LOL-B, are also lipidated, so they're anchored into the outer membrane, but the bulk of them sits in the periplasm, and this one, LOL-A, sits completely in the periplasm. We've thrown in, to make things interesting, we've thrown in this antibiotic, polymyxin B1. So this is, again, is an antibiotic we've looked at, studied in the past before, again with Pete Bond. So this antibiotic is thought to cause cell death by its action on the inner membrane, right? So to get to the inner membrane, it somehow has to go from the outer membrane across the periplasm to get to the inner membrane. So we threw whole bunches of this into the periplasm. And actually, not only are there proteins in these systems, but there's loads of smaller molecules, right? So there's these molecules known as osmolytes, where we can have, there's sort of ions, there's these osmoregulated glucans, there's urea, putrescine, spermidine. There's basically a whole ton of these small molecules. So we took realistic mixtures of these small molecules and added them into the mix. And before I go on to talk about what we found, I just wanted to give you a little bit of background onto two of the proteins we've looked at. And that's the this protein LOL-B and LOL-A. Now, just very briefly, the job of these proteins is to ensure that Braun's lipoprotein, and similar proteins get to their correct localization, get to their correct location. They're responsible for the localization. So LOL-A picks up bronze lipoprotein at the inner membrane. It carries it through the cell wall and delivers it to LOL-B, which is sat here just anchored to the outer membrane. LOL-B then somehow flips it to its correct location. The way this is achieved is bronze lipoprotein has got lipid regions. There's a hydrophobic cavity in both proteins where these lipids are carried. Okay, so the lipids bind to the hydrophobic cavity. You can just see here with simulations we did in the past, you can see bronze lipoprotein in yellow, and in space filling representation, you can see the lipid regions. And in both cases, you can see they've spontaneously bound into the hydrophobic cavity of LOL-A. Now, one thing we know from our simulations is that the cavity in LOL-A is fairly indiscriminate. Other hydrophobic molecules are shown in red here. This is an inhibitor molecule, can also bind into that cavity. Okay, so it's just a greasy cavity. Anything that's hydrophobic can bind into this cavity. We know that already. Okay, and that's important to keep in mind when I show the results that we have for these simulations. I'm just going to show you a gratuitously sort of pretty picture here where you can see a large system where we've got all these bronze lipoprotein is in purple. In green, you can see the antibiotic. Red is the cell wall, and yellow are all the other proteins. So the mixture we have has a realistic crowded volume. Okay, so this is the level of crowding one would expect to see in the periplasm in vivo. Whoops. Interestingly, what we see is that the antibiotic, which is also a lipopeptide, which means it has a lipid region, we find it also binds into the cavity of LOL-A. You can just see that here, and indeed it does so. When it does so, it binds to the same amino acids as we were seeing bronze lipoprotein binding to. Okay, so in hindsight, this makes sense. If you've got an antibiotic with a lipid region, why wouldn't it bind into an indiscriminate cavity of another protein that is designed to carry lipids? Right, it kind of makes sense, but as far as we know, no one has ever considered this for this particular antibiotic. And we saw it happen multiple times in our simulations. And what we're saying here is, could this be one way in which polymyxin can sometimes move across the periplasm? So we're not saying this is the mechanism by which the antibiotic moves across the periplasm, but we're saying, given the complexity of the systems, the sheer number of different interactions and the number of interaction partners, perhaps sometimes this does happen. Maybe and we're calling this hitchhiking, maybe this antibiotic hitchhikes on this protein sometimes. Oops. Okay, so to conclude, I think what I've shown here is that we found bronze lipoprotein plays a key role in maintaining the distance between the cell wall and the outer membrane. But we think, and that was known before. That I wouldn't, I'm not claiming that we are the first to say that. We saw it happen in our simulations, but experimentally this has been known too. But what we have found, which I think we're the first to say, is that it also plays a key role in facilitating the non-covalent interaction of other proteins in the cell wall, right? So we needed the bronze lipoprotein to be present for the monomer of OMP to bind to the cell wall. We've seen that TOL-R forms stable non-covalent binding interactions with the cell wall, and we've discovered various details of that. We think polymyxin B1 may be transported to the periplasm sometimes by hitchhiking on the lipoprotein carrier LOL-A. And I think the take-home message really from all of this is that the biological details of complexity of the individual molecules, but also their crowding, should not be neglected when we consider bacterial cell envelope systems, especially now given we have access to greater computer power and these bigger and more complex simulations are becoming increasingly feasible. And with that, and I said, we're working towards a virtual bacterial cell envelope model. So my plan ultimately would be to have one of these, such model at the atomistic level and at the coarse grain level. I haven't spoken to you about coarse grain stuff yet. Sorry, not yet. I didn't give you heart attacks. She's still going to go on and on. Today is what I mean. But it's a key part of what we do in the group, and ultimately they allow longer timescales. So the goal is to have the sort of horses for courses, really, the methods that are appropriate for the systems we're looking at. And finally, I'd like to finish by acknowledging various collaborators, in addition to the ones that I mentioned as I've been going along, various members of my group, sources of funding, computing resource, and to leave you with a picture of my group on a normal day at work. Thank you very much. I will stop talking.